So, children, the first chapter in biology that you have in class 9 is the fundamental unit of life. So, when we say fundamental, fundamental means basic or the most primary unit of life or of any organism is called cell. So, this chapter basically will deal with cell and the different aspects of cell. Now, little bit about cell you have got in class 8 also, so I'll go quickly through that. Now, the discovery of cell became possible only after the invention of the microscope. The primitive form of microscope was made by Anton von Leeuwenhoek, but the microscope which he formed or which he made was nothing more than a magnifying lens. Later on, Robert Hooke developed a compound microscope, and with the help of the microscope which he developed, he could discover cell. And you know how he discovered cell? He observed a thin slice of cork under. Cork is part of bark of trees. So he observed that thin slice of cork under the microscope and he found some empty box like structure. And those empty box like structure were given the name of cells. So before we go to the definition of cell and the cell theory, let us have a look at the structure of the microscope. Here what we have is a structure of a compound microscope, the microscope which you generally see in your laboratory. Now why it is called a compound microscope? Because such microscope is a combination of two lenses. One lens is on top, which is called the eyepiece. Other is here, which is called the objective lens. So eyepiece and objective lens, both the lenses are present. That's why it is called a compound microscope. This you can see, this is the body of the microscope. There is nothing but a hollow tube. This wheel that we have here, this is called the coarse adjustment. Coarse adjustment, if you move it, if you rotate it, this body moves up and down. Then we have a fine adjustment. After focusing the object, we have to bring the object much clearer. We can make it much clearer with the help of fine adjustment. This is with the help of which we hold the microscope. This is called the arm of the microscope. Now you can tilt the entire microscope with the help of a swivel or a screw to which this arm is mounted. Now below the objective lens you have the stage. On this stage we put the slide and the slide is fixed with the help of two clips. Just below the stage we have a condenser. Now how a condenser looks like, this condenser helps in controlling the amount of light which is passing through the microscope. A condenser, it looks like this. From below there is a handle here. If you move it up and down, this aperture, this opening will become either broader or smaller. So if it becomes broader, more light will enter, and if you turn it like this, it will become smaller, so you can regulate the amount of light. Now below the condenser is a concave mirror. On one side it is concave, one side it is plane. So with the help of this mirror you can make the light pass through the specimen and through the tube. And this entire setup, it rests on a foot. So this is the overall structure of a typical compound microscope. Now on every lenses you'll find power written. Suppose 10x is written here. So 10x, you know, 10 times multiplied. Suppose here it is written 10x, here it is written 10x. So what will be the total magnifying power of the microscope? It will be 10 into 10, 100x, 100 times magnified. It is not 20x. So don't make that mistake. How to find out the magnifying power? So after Robert Hooke discovered the cell in the year 1665, a lot of scientists, they researched on different organisms, and ultimately in the year 1839 and 1838, two scientists, Theodore Schwann and Matthias Claydon, they gave the cell theory. Separately they gave, and both the cell theory, the main point was that cells are the structural and functional unit of life. That means any living organisms, ultimately their structure will be made up of cells. So right from bacteria up to blue whale, all living organisms on this planet Earth are made up of cells. Second point was that cells are the functional unit of life. That means whatever function goes on in an organism is due to presence of cell. So this cell theory was later on modified by another scientist, Rudolf Virchow, in the year 1855. According to him, because we know multicellular organisms are there, so from where so many cells come? All the cells comes from pre-existing cells. So new cells arise from pre-existing cells. So this is the cell theory and the modification. Now if you look at the cells, cells are generally microscopic, except for some big size cells like the eggs. Most of the cells are microscopic, and size of the cell is measured in a unit which is called micron. So when we talk about micron, it is represented by the letter mu. So 1 micron or 1 micrometer is equal to 10 to the power minus 6 meter. 10 to the power minus 6 means 1 by 10 to the power 6 meter. 1 by 10 to the power 6 means 1 meter. If we divide it into 1 million parts, out of 1 million part, 1 part will be 1 micrometer. So small is the unit of measurement used for measuring the dimensions of cell. So that is how size of cell is measured. If you look at shape of cells, cells come in varied shape. Cells may be spherical, cells may be flat tiles like, cells may be cubical, cells may be columnar, brick like, cells may be your rod shape, they may be cylindrical, they may be spiral in shape. Then we may have comma shaped cells. Cells may be discoid. Discoid means just like a pizza base. Red blood cells. Red blood cells are the smallest cells of our body. So this shape is called discoid. So cells may be discoid in shape. Then you may have wire-like cells. Example in case of nerve cells. Cells may be shapeless as in case of amoeba. So there are different types of cells. Now shape of the cells is determined by their function. Whatever function they have to perform, accordingly the shape of the cells have evolved. For example, red blood cells, their main function is transportation. Therefore they are discoid in shape so that easily they can flow. Then the cells of our skin, these are flat tiles like cells because they have to fit very closely with each other. If they are spherical, there will be gap between them. So they are flat tiles like cells. Similarly, nerve cells, they have to carry information, therefore they are long wire like cells. The largest cells is the egg. Egg is regarded, technically it is regarded as a cell. So obviously ostrich egg is the largest cell, and red blood cell is one of the smallest cell present in our body. So overall, this is the initial concept of what is cell theory, how a cell looks like, what is the unit for measurement of cell size, and how we can view a cell with the help of a microscope. We place the slide here, we adjust the mirror, we adjust the adjustment, and we view with the eyepiece out here. So overall, this is the introductory part. In the next video, I'll be discussing about plasma membrane and different type of transportation which takes place across plasma membrane. Thank you.